Davis?
My PhD is in horticulture and agronomy, but my emphasis is in weed science. And my lab actually specializes in weeds in rice. And so we usually see, if you know Dr. Eric Webster, I think it's at
Yes.
LSU. Yeah. So we usually, we see him.
And who do you work with at UC Davis? Sorry. Yeah.
I work for Qasim Al-Khatib.
Yeah, I think I remember the name because maybe I met him in one of the RTWG meetings, you know, the Rice Technical Working Group meetings. Yeah, and probably one at California it was. Yeah.
Yeah, the one in Prague. Oh gosh, my colleague Kaio Brunharo, who is now a professor in Oregon. Yeah, he went to that meeting.
Okay. Yeah, it was in Long Beach, correct. Yeah.
Yeah.
Okay. And you are in Oklahoma where now? Stillwater?
No, I'm taking care of my parents. They live up north of Tulsa. Yeah, close to Tulsa. Yeah.
Yeah, I drove all the way to Oklahoma last summer. I remember I had to bring my kids, and then I have a couple of friends. They invited in Nobel Foundation, Nobel Research Institute now. And Ardmore, yeah.
Yeah. I would really like to work there when I graduate, but I was told that the Noble Oil was purchased by Texaco, and so I'm not sure how long that they will be the Noble Research Foundation.
Oh wow!
That's the rumor.
Things are changing fast. Correct.
Yeah.
They used to be a foundation. Now they are kind of a private entity, Nobel Research Institute (LLC). So. They have a bunch of, I think, administrative changes.
Yeah.
Okay, nice. When I was telling to Subhabrata that it's so nice that you people are young, energetic, and then organizing this one free. Some many students and professors like there or researchers, they are just getting information free. Even though I remember from Subha's email, you go by Subha or Subhabrata, everybody has to tell the full name. Yeah. Unfortunately, I do not have a nickname, so people had a difficulty originally. When I went to Philippines first, they said, "Can we call you John?" I said, "I'm actually a Hindu, so I don't know if that name." So, but anyway, so yeah, so it's a very good platform and then very good initiative. So I thought it's neat. So I'm very awkward with this webinar and this online presentations. I don't know. I'm probably an old school guy, but I will try my best to keep up with. So
we are there for you, sir.
Yeah. Okay. And that's the spirit. That's good.
And we are now online, and I'm waiting for 2 or 5 minutes. Now 145 people are watching. So I'm waiting for 2 or 3 minutes. Then Liberty will introduce you. I am collecting the questions. Neha also tried to collect the questions, and then we make a list of questions for you. And we put this question in the chat box. There is an option. Just the button share screen is chat. So when we paste it, it's blink. Then you can choose a few of them, not all. But I try to collect all the question and mail to you after the webinar. I emailed you the appreciation and the comment and the questions. And
I know it's a very awkward timing for probably many.
Yes. It's a little bit late, but I am interested in that time because many people are watching in other continents. So it's my intention that we try to shift our timing in second half, because so many African countries are watching our webinars. So we got about 56 or 56 or 51 to 56 countries for today's webinar registrations. So then there after the question answer round, there is a short interview for you. These are the one type of motivational, because we know your profile, but we try to know something which is not available in the such profiles or academically available, but is yes, and it is give us
It can go for days, no? No, I'm just
Oh, that would be nice.
no. You tell for every day for 1 year of my age, you know, my life. So no, I'm just.
Let's start a series with that. All right.
Just I need to just yes, Neha, you can you try to say something? Actually, I missed.
No, I was not saying anything much, but it's good to see you in lab. Is the lockdown?
Oh, okay. Yes, I'm just joining my lab 3 days back.
That's good.
Okay, because it's a long time that my background is same.
So you are allowed to do so now. Restrictions are over?
Not over, but I need some official work related to project submission, and that's all. So I have to come. But there is now in West Bengal there's a festive season going to start, Durga Puja. So it's a short window that I can use to come in my lab.
Okay, that's good.
Is the lab shared by both Madam Karavi and then Swapandata?
Yes. It actually both are the PI and co-PI for the DBT funded project, and Sir has a special project, and I'm the only resource person for that project. It has one RA post, and I'm doing that. So our lab is funded by DBT, and it's in Calcutta University, Baligan Circular Road. Okay. So, sir, we are ready. We are ready. We are ready in YouTube. We are ready in Facebook and LinkedIn.
Okay.
So I request Liberty. Go for the Liberty first. Then I'm come to the question answer round. Neha, you collect the questions and edit. We all collect the questions and try to edit. And Soma, you have the interview questions, and that time you can go for the interview. Okay, let's start. Okay, so I just go to my full screen. Speaker. Hello. Okay. Welcome to Bioingene webinar series. This is our 19th webinar. We are very honored and grateful that speaker has accepted our invitation from this humble platform. We are very fortunate to have you, sir, to this platform. I'm request Liberty, my friend from UC Davis, to introduce today our speaker for today's webinar. Liberty.
Thank you, Shiva. Welcome everyone to the Bioingene webinar series. Today is our 19th webinar of the series. Bioingene is a nonprofit organization created to promote plant research worldwide. We're very happy to organize plant research webinars that are free to the whole world. The webinar series has been designed to build a platform from where plant scientists can present their research to the world. We hope future scientists can gain perspective and inspiration by listening to the esteemed plant researchers talk about their scientific accomplishments and their thoughts on the future of plant sciences. We are grateful that many renowned scientists have accepted our invitation to share their research insights with us, and we hope that you'll join us for future webinars. For today's webinar, we have received a huge response from 51 countries all across the world. We welcome all the viewers and thank them for their interest in Bioingene. You can apply for a certificate of participation via the feedback link and password, which is provided after the Q&A round, which will occur after the presentation. Submission of the feedback form is mandatory to receive a certificate. The feedback link will be provided in the YouTube chat box by the host. Please be patient. You will get ample time to fill and submit your feedback form. Please collect your participation certificates after 2 days from the end of this webinar. You can visit our website to register for upcoming webinars. You can connect with Bioingene via Facebook, YouTube, Twitter, and LinkedIn. The topic for today's webinar is Harnessing Transcriptome Resources of Non-Crop Models for Improving Stress Resilience in Rice by Dr. Niranjan Baisakh. Dr. Baisakh is an associate professor at Louisiana State University under the School of Plant, Environmental, and Soil Sciences in the United States. He did his graduate and postgraduate in agriculture, specializing in plant breeding and genetics. He did his PhD in plant biotechnology at the International Rice Research Institute in the Philippines. After his PhD, he worked as a visiting scientist at the Temasek Life Science Laboratory in Singapore. He worked as a project scientist at the International Rice Research Institute in the Philippines and later joined Louisiana State in the United States in 2000. He is a specialist on plant abiotic stress, molecular biology, functional genomics, genetic engineering, tissue culture, and dihaploid breeding. Dr. Baisakh has received many grants and awards for his contribution in research. He received the Service Award for serving in the Louisiana State University Interinstitutional Biological and Recombinant DNA Safety Committee in Louisiana in 2014, the Teacher Merit Honor Roll, Louisiana State Chapter of Gamma Sigma Delta in 2014, the Louisiana State Campus-Wide First Laboratory Safety Accreditation Award in 2011, and the Tipton Team Research Award from Louisiana State in 2007 and 2019. Dr. Baisakh has supervised more than 50 researchers, including students, postdocs, visiting scientists, and short-term trainees. He has published a large number of articles in international journals. He is on the editorial board for reputed international journals like Scientific Reports, PLOS ONE, and he has just joined the Crop Science Journal. We are highly honored to have him with us today. I now request that Dr. Baisakh please share his screen so that he can begin his talk.
Okay. All right.
Yes, sir.
Yep.
Yes, it's visible.
Okay. So, I thank you, Liberty, for that nice introduction, and I would like to also thank Bioingene group, the team, for this initiative, and also, reaching out to me and asking me to share some of our research that we have been doing in my lab at Louisiana State University Ag Center or Agricultural Center. Okay, so today I will talk about a little bit different things. Maybe, I don't know. I will be talking about non-crop models. And I will come to that one. We all now, before I go into the details of my talk, first let me tell you that this is where I am at. So I'm at Louisiana. And if you look at the map on the left, this is actually the Louisiana crop map. And I'm actually blessed that I'm working in a state where you can find almost every crop. Okay, so the climate is like that, that it suits to rice, and you can grow rice, sugarcane, corn, cotton, soybean, wheat. So anything that is going from subtropical to almost temperate, you can grow over there. And unfortunately, LSU Ag Center being the flagship at the Louisiana State Experimental Station, so we have regional research stations in almost all parts of the Louisiana, dealing with individual crops. I'm not talking about the dairy or the cattle industry or the ornamentals and then the fruit crop or vegetable industry here. I'm talking about mostly the field crops. Okay, so, and I am also very, very lucky to be working on primarily two most important commodity crops of Louisiana, that is rice and sugarcane. Okay, So this is the buzzword, right? Climate change. So you believe it or not, climate is changing, and climate is changing since we were born or from time immemorial. So, but yes, we are contributing to climate change. That is definitely not undeniable. But there are two different schools of thought with this climate change, right? So the key culprit of this climate change is actually carbon dioxide, right? The carbon emission. So there is also a school of thought which thinks that the carbon is actually contributing to the crop yield by enhancing the photosynthesis. Okay, so that you can see over here. Let me go to that pointer option so that I can put for a little bit. Yeah. So the green ones that you can see here, it's probably the ones that thinking that carbon is contributing to the increase in yield. But there are scientific reports are still kind of dirt that you would know whether it is really true. And if you look at the other part of the map, you can see that there is significant loss in the crop yield worldwide due to this carbon emission caused by the climate change or causing the climate change or global warming. So what are actually the impact of this climate change on rice, particularly since my talk is focused on rice? Um, Altered timing and magnitude of precipitation, that is actually can induce either drought or flood. If it is too much water, flooding, and too little water, the drought. And of course there is global warming, so there is this increased temperature, and this can actually have also impact on the disease and insect occurrence in rice, right? And in addition to that, the enhanced UV and increased temperature also can alter the competition between the rice and major wheats, and also some of the beneficial bacteria in the soil, including the nitrogen fixing bacteria and all other beneficial bacteria. So ultimately all these contribute to the or affect the rice growth and productivity. So so that says that we have increased pressure for more rice production to feed the world, and then the rice consuming population in the world is increasing. I'm not going into the statistics or the data. Most of the slides I will actually try to avoid the data. They are ever changing. You can Google, you can get a lot of information about all of these things. So if you look at just typically, I would be focusing my topic on salt and drought. So if you look that this is actually the global map where these patches show that how the salinity is kind of worldwide. It's not just like specific to one particular area, but it's actually worldwide. And in the United States also, you can see here, these are some of the patches where either saline or sodic or alkaline soils, So this is again caused or aggravated by the climate change. And I'll talk a little bit when I come to this Louisiana thing. So here in Louisiana, we have a prediction that 40% of the rice yield loss is caused by salinity. And I do not know if all of you are aware that Louisiana, we are in the coastal south. So every time we are actually kind of encountered with hurricanes, And in fact, as we speak, we are preparing for a hurricane delta. We just had a Hurricane Laura a few weeks back, and then now we are preparing for another Hurricane Delta. So what these hurricanes do, in addition to all this damage that does to the humankind, it also affects to the field, the crop field, So every time there is a hurricane, the seawater actually inundates into the land and then makes the land saline. So for example, field crops like rice and others, they cannot grow in saline soil. So this is also contributing to due to the climate change, this is also being aggravated, and we have this problem even in Louisiana here. And I'm talking about, although these are global problems, but my talks will be a little bit centered in my research will be more centered towards what we do here at Louisiana. As a state employee, my mandate is actually to work towards the improvement of the livelihood of the people, people of Louisiana. So this salinity is actually one of the major problems in Louisiana. And similar is the case with drought. I think all of you are aware, there is no need to talk about how important drought is for rice. Drought in many different stages can affect the growth, and then also in the reproductive stage that we call a terminal drought, it can actually significantly reduce the yield, can cause to even total failure of crop yield in rice if it is a prolonged period of drought during the flowering time. So there is this study from IRRI, a prediction that by 2025, almost 15 to 20 million hectares will suffer a varying degree of water stress. You can see that it's widespread. Okay. In US Drought Monitor, so in US, most of our crops are irrigated, but we all know that irrigation is actually expensive. Pumping water is expensive, and surface water is actually not very good nowadays. As I told you, there is of course these hurricanes that pollute the water by alkalinity or salinity. And also, when there is salinity, even if we have a lot of water, we also experience drought because we call it as a physiological drought, right? So when the plant, the plant has enough water in the soil, but still it cannot really take up because there is too much of salt in the soil water, or the surface water. So this is actually a monitor which actually gives you an idea about how the dry areas or the areas with dry period progresses. So we are here in Louisiana, and here sometimes you will see that we will experience some unusually dry periods. In the next slide, you can see here, this is actually a snapshot from April through August, because these are the 5 critical time that rice is grown in Louisiana. So starting from April, you can see here, rice is grown, if you remember my first slide, that rice is mostly in the southwest region, and recently it is moving towards the north. I will talk to it a little bit later. Between April and August, you can still see that there are actually dry spells, especially in the rice growing area. In June is the month where we experience more rain. So that's the reason you don't see that, but for this particular year. But then we do have periodic dry spells, and the farmers need to actually pump irrigation water. And rice is primarily cultivated under flooded condition in here. But we know also that the flooded field contribute more towards global warming through methane gas emission, right? So this is actually kind of stress. So we know that this is really stress, right? So I actually stole from some site in Google these images. All forms of life have evolved strategies to adapt to environmental stresses, right? So whether it is escape, or you avoid it, or you tolerate it, So the plants do have similar strategies at the organelle or the tissue or the cell level. So how the plants actually adapt, because we do know that there are variations that exist actually in primary gene pool. For example, in rice, this is actually a picture that is from the IRRI website. This is one of the lines developed by IRRI. It's an integration line with Pokali. and this is an IR29, one of the first varieties of IRRI. So it is very highly salt sensitive, whereas this one looks pretty much good under the hydroponics salt condition. And same thing also, we did have actually a lot of rice in the diversity panel that was developed by Susan McWoods and others. So we screened actually a lot of those varieties, 420 varieties in the greenhouse condition. These are the two varieties that we see here, the two Indian varieties, Vandana and N22. They kind of, at the vegetative stage, they kind of maintain still the relative water content, or they are show the tissue tolerance, and this is one of the US varieties. I'm just showing this snapshot because we have developed populations using these varieties of rice. So the bottom line is that there are, of course, variations existing in the primary gene pool of rice. And this is actually very common for all crop improvement program. First of all, you are looking at the germplasm or the genetic diversity. And then once you have that one, then you can go different routes. So you go for developing population, biparental population, or diverse panel, and you go for either biparental QTL mapping or from the diverse panel genome-wide association mapping, or you go to the transcriptome and the functional screen of this transcriptome. Okay, so today I will be focusing on this particular aspect. So how we'll be or we are using transcriptome and functional screen to identify the candidate genes, and then for their downstream application in gene manipulation, such as genetic engineering or gene editing. Okay, we know there are variations in the primary gene pool. If you see, there have been exemplary success in development of drought-tolerant rice varieties at IRRI and elsewhere. But this development of climate-resilient crops through conventional breeding has been slow. That is not because of anybody's fault, but it is per se because of the low heritability of the resistance traits. At the same time, also the complexity of the resistance, because when we call about drought tolerance or drought resistance, we are talking about either genes involved in avoidance or in escape or in tolerance. So there are a lot of genes involved, and because of this quantitative nature of the resistance, so the conventional breeding has been slow. Nevertheless, there have been significant progress, as I said. I know a few varieties have been released by the International Rice Research Institute and elsewhere too. And I very much like when Dr. Jawahar Ali from IRRI, he actually presents in San Diego his green super rice. I simply I'm amazed that there are rice varieties which are actually having many different traits put together. So yes, there have been some success, and even in salinity, I know the CSR 27 and some IRRI lines have shown promise in saline conditions. Salinity is a very tricky area because you are looking at a plant completing its life cycle to yield under constant salinity, right? So it's not like a drought. Sometimes drought could be just a periodic or maybe a spell, but for salinity you are dealing with a field which is constantly under saline conditions. So that is where probably the problem is that there has not been much success at the development of salt tolerant rice varieties. And that's my thought. So to kind of cape up here, but because irrigation water is becoming expensive, water is not anymore considered as a renewable natural resource that you can just have plenty and then anytime And then either if it is not available or if it is available, then it is poor quality. And then also if it is available, then pumping is very expensive, and we know many places the aquifers are going down, getting low. So that's the reason farmers have actually, and then scientists and researchers have adopted these strategies. I know alternate wetting and drying. So these are some of the concepts IRRI and then other countries, they actually have started to adopt, where actually you can keep the field not flooded, but you just flood it and then let it dry. And then when it is dry, then again you flood. So you don't keep the field on constant flooding, and here, so you can save some water over here. And by this, you are not really having a lot of yield penalty. And another approach is what we call row rice, or growing the rice in rows, just like you grow any other row crop, like soybean or corn or cotton. So here you are actually growing the rice in rows and then keep the water in furrows as and when you need. And in Louisiana, for example, in the north part of Louisiana, we are actually adopting more of this row rice because that the soil in Louisiana, the north part of Louisiana, is pretty hard pen. So even if it rains, the water actually does not stay for a long time. So there are farmers, more and more farmers are actually getting interested in this row rice, around 8,000 acres in Louisiana itself. So I know it is actually going to be more and more popular across the world. I saw the other day, China, for example, they are experimenting, or some companies experimenting drip irrigation system. You don't need to really have rice flooded, but then you can have drip irrigation, or you can have semi-aerobic, which is pretty much kind of drought. Okay, so you do not really keep it under flooded condition, but you only irrigate the field only when you have some kind of sensitive symptom like leaf rolling or plant drying. Okay, so this is actually some of the work that we have been doing here at LSU. Some of our lines that I told you that we have developed, and we are screening under the semi-aerobic condition in Texas, Vermont. And we have identified a few promising lines that we are actually purifying, and then that would go for. So these fields have never been flooded. They have been always kept dry. Only when we see some symptoms of severe drought, symptoms like leaf drying, or plant is almost getting to the point of permanent wilting, then there will be some flash irrigation, just to keep the soil saturated. Okay, so we have been doing some work, but then the question is, then why extremophiles, right? So you would essentially want actually a plant which would have all these traits. Okay, if you want to develop a rice plant which is salt tolerant, you would want to have it exclude the salt at the root level. It should selectively uptake the potassium ion and then exclude the toxic sodium ion. And even if it takes off more sodium, then it should have a mechanism to exude the sodium or excess salt through, either leaf glands, in the glands in the leaf, or something like that. And then even if there is still sodium ion, then they should have the ability to sequester the toxic sodium ion into the vacuole. Okay, so it's like ion compartmentalization and sequestration, vacuolar sequestration. And the plant should have the ability to accumulate compatible solutes like inositol, glycine, betaine, choline. So those things actually maintain the cellular homeostasis by actually maintaining the osmotic potential of the cells, right? So these extremophils, they actually have all of these traits to survive under extreme weather conditions. So some of them are what we call them halophytes, because they kind of like actually saline water for completing their life cycle. So you might be aware of Thelungiella and Mesembryanthemum. These are the two two halophytes that have been very widely studied. And so also Sweda, Salicornia, and then this is a mangrove, Avicennia, and a triplex. Okay, so these are some of the halophytes that have been very widely studied. Unfortunately, all of these are actually dicots. Okay, so there are very few monocot halophytes that have been studied much detail in greater detail. Today, I will be talking about some of the work that we have been doing in Spartina alterniflora. It's actually a smooth cord grass, and Potatia quartata. It's actually a wild rice. So these are actually monocots. Because our objective is to improve rice, which is a monocot. So we actually thought that it's probably more worth to look at the monocot halophytes because the mechanism of salt tolerance in the monocot halophyte, it might be easier to look at these mechanisms and try to install that in rice or translate those information into rice. Okay, so coming to Spartina alterniflora, this is called smooth cord grass, and it's native to Louisiana. It's actually considered a weed species in California, in Los Angeles and Bay. It's considered a weed because it's very vigorous, rhizomatous. And if you put a plant today here, probably in 15 days, 20 days or a month, you can see a plant almost like 10 feet away, so that is how rhizomatous and then aggressive it is. But this is actually used for our coastal restoration because of this rhizomatous nature, and also high salt tolerance, because they are growing in the coastal wetland, and then every time there is salt water splashing over it, but then these plants are still standing. And they survive, and then they produce seeds. So, and they physiologically, it has been already known that it contains all those possible mechanisms to tolerate salt. Genetically, it's a very complex plant. It's a hexaploid, and it's actually a NU hexaploid. It's like 2N is 62. And then the genome size is around 1.8 GB, which is kind of around 4 to 5 times more than rice, So it's not really an easy plant to work with in terms of its genetics because of its annual polyploid condition. We actually kind of proposed this one as one of the monocot halophyte model for rice salt stress improvement or salt stress response improvement. Similarly, Patria, this is actually very near and dear to me because I come from India, from a place where this is actually, you can find the primary center of the origin of this one. Although, Eastern India is actually the place where you will find this in mangroves, in salt marshes, and it is the only wild relative of rice, which is an halophyte, And although I just saw maybe a couple of years before that, there has been a study from IRRI that Oryza alta and Oryza latifolia, these two also show considerable degree of salt tolerance that we probably need to look at. But this is actually again, like this also is known to have all possible mechanisms of salt tolerance. It's a tetraploid and it's a wild relative of rice. And there have been transcriptome assembly of this published from India, and also a draft genome, very rough draft genome published also from India that covers around 85% of its genome. But I am aware that Broadwing's group in University of Arizona has platinum standard genome assembly, and we also have a very comprehensive transcriptome atlas of this at a different salt level, different tissue that is actually under our study now, and then we have not published that. So this is actually very important because it's a wild rice, So this particular one was used for making crosses despite several failures, again, a scientist called Dr. Kiroz Jena from IRRI, he was successful in making an interspecific cross with this, Oryza quartata with rice, diploid rice. And so I'm pretty sure that they are forwarding. I have seen some news that they have actually gone to develop some backcross inbred lines, and then screening for salt tolerance. And then I'm pretty sure they must have done more and more progress on that front. Okay, but before working on this one, my laboratory was more focused on this sputina. So when I actually started in 2004 at LSU, we did not have much of molecular information on this grass. So I was intrigued by its salt tolerance ability. So we were looking at to look at some genes that would be very highly responsive to salt stress in this particular grass. Although my job was also to look at the genetic diversity of this particular grass in the coastal states, in the coastal state of Louisiana. So there are several different accessions. So we used to collect and then do genetic fingerprint. So, but then because I was interested, so at that time, we did not have the high throughput sequencing technologies. So we used to still go. I used a technique called cDNA FLB, which is very small scale. Because whenever you deal with something with gel, so you cannot really think of in hundreds and millions and thousands. So we did, followed by that, we actually did a cDNA library of both leaf and root from this particular grass. So those cDNA were actually the one after actually exposing these plants into the salt stress. So they are salt-induced transcriptome, but mostly the cDNA library was prepared. And we actually identified a few of these ESTs which are very responsive to salt stress, whether it is in the leaf or in the root. Some of them most of these are actually published work, as I said. So, I think Subho actually made me very clear that I should not be putting anything that is not published, since it is very open. And so this was actually kind of a very pilot scale or preliminary study of the biology, understanding the biology of salt tolerance of this grass. And then we actually leveraged this information to secure some funding, and then went for the genome-wide scan of that particular grass for the transcriptome profile. So originally we had a 454, that was actually the first generation sequencing, that is pyrosequencing by 454. And then later on we had added, in 2018, we added actually the RNA-seq data also. So we have actually the 6x coverage of that grass transcriptome. And these are actually again, like these are the data. I'm not going into the details. We have now, this is actually the leaf, the transcriptome coming from the leaf, the unigenes, leaf under control and leaf under salt stress, and then this is root under control and root under salt stress. So we have a lot of unigenes that are actually common among between the two tissues, and with and without stress, but we are interested actually the ones that are specific to the tissues and under a particular stress, under the salt stress. So whenever you have actually a RNA-seq or next-gen sequencing, you will get all this huge number of unigenes. And then what you do next? Okay. Before going to that, so again, this is a proof that using some of the genes, we know that this is a grass, so it should be actually close to the grass in the tree. And this is here sputina, which is close to corn, GMS, and rice, and there is one xerophyte viscosa, and potassium is over here. So as you can see that this sputina and potassium, these two are actually kind of clubbing together over here. Okay, and then in the same clade is in barley and then oats. And then these are, this is, the wheat is over here, even though it's a grass, but then in terms of this genes for the salt tolerance, and you know that barley has also a lot of salt tolerance. So, and this is actually the other clade that contains these things. And Sueda, even though it is actually a halophyte or Mesembrothymium abyssinium, because they are diploid, so obviously they have been in a different in the clade. And these are actually the genes of Spartina. They are actually distributed across the rice chromosome. You can see here. So these are the genes that are distributed across the rice 12 rice chromosomes. Okay, so obviously there are some gaps, and we do have, this was actually made with only the 454, but these gaps we have closed down with the RNA-seq data, and then I have not included here that we have not published. Okay, and then going into the genes, then the objective is, when you have this next gen sequencing, the objective is actually to narrow down the genes that you really want to target. Right, that's actually your number one objective, that because you cannot possibly handle that many thousands of genes. So, and then you go for a gene ontology enrichment of these genes, and then you identify these genes or the processes which are actually enriched under salt stress. So, for example, here we have kinases and the genes involved in the transport mechanism, and the transcription factor like DNA binding or protein binding, and the genes in transferase activity or metabolic processes. So what the bigger the circle, that means more number of genes, and then the darker the color means they are highly enriched. Okay, so under salt stress. So we are actually targeting, that means this is a kind of narrowing down of the genes, that you only select the genes that are in these pathways because they are more enriched under salt. And we actually divided that into leaf. I'm not again going into the details. So here, for example, under the biological process, you have more into the genes in response to stress, which is very expected. And again, in transport mechanism, those which are in the plasma membrane, we know that transmembrane genes with transmembrane activity or present in the plasma membrane are very important, because there are a lot of literature that actually suggests to that. So the same thing also for root. Again, I'm not going into the details of this. These are all published. But again, here also, there are also these genes that is very highlighted. They are actually more enriched in root cells also. Okay, so now we have these genes. So we needed to actually see their differential expression under salt. So the ones that you see here, the green dots over here, this is actually MA plot where you see the genes that are actually with the green dots, green circles. They are actually having log 2 fold over 2, which is, that means more than 4-fold increase under salt stress. Okay, and then the red ones, the ones that are actually down regulated, that means less than or 4-fold decrease in their expression. And this is actually just a snapshot of one of the couple of categories of this. This is actually the kinases, and these are the transporter genes. So as you can see that the blue ones, of course, are the upregulated, and the red ones are down regulated. And then with the hierarchical clustering, this clustering also helps us in, again, selecting out some genes or narrowing down the number of genes that we want to target later. Okay, so this is a validation of the genes, that what we actually see in next gen sequencing, they are actually validated when we really do the temporal expression of those genes in both leaf and root tissues over different time points, very early to until 3 days. So we can see here actually there are differential expression, and some of the genes are maintaining their expression even at very late stages of salt stress. So those are some of the genes that we are actually also interested, because you obviously want to have a gene which is expressing, not only just expressing in the early stage. And some genes can express just because of a shock. But then the maintenance of the expression of the genes is very important, because then that these genes are actually truly responsive to salt stress, or they might be actually contributing towards the salt tolerance of this particular grass for their prolonged expression of those genes. Okay, and this is again the real-time PCR of some of the selected genes. In addition to the transcriptome, we also look at the small RNA profile, so that to look at the post-transcriptional regulation of the salt response of this particular grass, Spartina. And this is actually the data that was actually in 2015. So this has definitely changed, and I should have changed actually. That there are a lot of microRNAs that have been loaded after 2015 until now in the Sanger database, and as I just learned from Nia that the microRNA 820 is only specific to rice, and then might be something in here we don't know. So this is something that we need to look again. But then we do have the microRNA profile of this grass, and then we did a microarray of this microRNA using a commercial facility. And these two microRNA, 397 and 528, actually stood out in their response to salt stress. Okay, and what we did, we actually selected out some of the known microRNAs that are reported in literature to be responsive to salt stress, and these two microRNAs that we identified through our microarray study. And also, so we looked at actually their conservation, among these members, for example, this is in Spartina alterniflora, and then this is in rice, and in corn, and in brassica and Arabidopsis. So we see a good bit of conservation of these small RNA of this grass with rice and other crops. And the ones that we actually saw, we selected out and we looked at their expression. These are the small RNA, their expression in both leaf and root tissues, and also their targets, corresponding targets. Okay, so the corresponding target of 397 here, a corresponding target of the 528. So we looked at the expression of the microRNAs as well as their mRNA targets, so that we can identify a microRNA mRNA module which is more responsive or more operational under salt stress in Spartina alterniflora. And we have overexpressed one of the genes, one of the microRNAs, 397, and we did not see a lot of changes with respect to their tolerance in the very first. But when we actually put this wild type, which was struggling to survive, and then back into normal water, regular water without salt, then the wild type could not make out. It is kind of died, wilted and died. Whereas the salt 397 overexpressors kind of revived and then started to grow. Okay, so this is actually the strategy that we followed with Patria, because we thought like this is maybe a straightforward approach to identify the genes or to capture the genes that are more direct. So this is actually a strategy, gene hunting. This is not a new technology. It has been used by several groups in several different crops, but not in many plants though. There have been few reports in plants, but mostly at bacterial level or at yeast level. So we actually adapted a system called COX or FOX, like full length overexpression strategy. So this is actually a functional screening. What you do here is you take the total RNA and then you make the cDNA, double-stranded cDNA, and the full-length cDNA, and then put the cDNA library into actually a vector of your choice, a plant transformation vector, in this case a PMDC. And then you actually make the agrobacterium library with this cDNA library, and the agrobacterium you transform with Arabidopsis or to rice. So this is actually we call gene hunting via functional screening. Okay, so using this strategy, we screened like millions of Arabidopsis seeds. We developed more than 1,000 Arabidopsis transgenics, thousands of Arabidopsis transgenics, and then we actually plated them in salt medium along with the hygromycin selection. So whatever made from here, so these are actually putative salt tolerant plants, and then we actually again in the next generation we picked up these plants, and then in the next generation we again grew them in the plate. Okay, so we grew them in the plate, and we selected out whichever, because some of them were probably escapes, they could not make it, but most of them actually again survived in salt supplemented media. Okay, and then it's whatever survived in the plate, we actually took them into the pot and then again put salt stress. So here you can see that this is your wild type, and then under normal condition they are all growing fine, and when it is salt, then there is obviously growth is very retarded, or growth is affected, but compared to wild type, these plants are looking much better. Okay, and then when we continued that salt stress to the reproductive stage, you can see here that these are some of the lines that actually successfully produced seeds. Okay, so again, like we did a lot of those physiological studies with these plants, looking at their root growth, and then the root biomass, and then the shoot biomass, and the root length, and the relative water content and membrane stability for the the tissue tolerance, at the same time the ion leakage, and also the um, um, studying the oxygen radicals, or by hydrogen, the hydrogen peroxide or the free oxygen radicals using the DAB or NBTSA. So wherever you see here, wherever you see dark coloration, that means the wild type, for example, here the dark coloration shows that the wild types are sensitive, and then producing a lot of those free oxygen radicals. At the same time, the transgenics are not showing that much of coloration compared to the wild type. So the same strategy we used for rice. Of course, in rice we did not have the luxury of producing thousands of independent transgenics, but we did have quite sizable number of transgenics. So in rice we screen them first, and we screen them in float leaf disc assay, and then whatever survived, then we actually also did screen them in the seedling stage. So you can see here, these are the trays that have independent lines of rice, and under hydroponics with 150 millimolar sodium chloride, and then whatever survived, we again went to the next generation to confirm that they are salt tolerant. So you can see here the clear-cut distinction between the wild types here, and then the lines, the rice lines that are overexpressing the Patria quartata genes. And again, these are the same physiological and biochemical studies we did to look at the tissue tolerance, and then ion permeability, and also the free oxygen radicals and superoxides. So we know that the extremophiles not only just tolerate salt, but they do also have the ability to tolerate a drought, because I think I explained before that even though there is a lot of water, they are growing in a lot of water, but then there is actually a physiological drought because of the salinity in the water. So we also tested these plants for drought stress, and we can see clearly there are few lines that are very, very good in drought tolerance, and then the next generation, when we go, we could see that the wild types hardly could make up after the 2 weeks of drought or no watering, and then some of the lines actually have survived the water stress. Okay, so how am I doing with the time, Shubho? Is it
it's okay, sir. You have a time for.
Okay, all right. So I mean, yeah, I don't know, maybe around, okay. So, and then once we have these genes, for example, the genes that we have, again, you need to always narrow down. The basic idea is actually to narrow down to focus on only a handful of genes. So we have actually here some genes that we saw for Arabidopsis, for example. The rice one I have not listed here. Again, this is under review, not published. So we actually we took the genes that were in Arabidopsis, in rice, both under salt and drought, and then looked at the interaction among those genes. So it's only those genes interacting among each other. So, and then we saw that these are the genes, some of them I have just dragged them, that these are the genes that have maximum interaction with the other candidate genes. So that actually shows that we probably could target these genes, and ultimately will be perturbing the expression of the other genes that these are interacting with. So that is actually the idea, and we would be pursuing that work. So once we have the genes, we know the genes, for example, in Spartina, so we need to actually functionally validate them, right? So you make a bunch of transformation constructs or overexpressor constructs, and put them into either model plants like Arabidopsis or tobacco or rice. So we chose several genes. I have just given the ones that we have published so far. For example, a case study of vacuolar ATPase. I told you that the plant to be salt tolerant has to have the ability to sequester ion to the vacuole, the sodium ion to the vacuole. And this is actually, the ATPase is actually one of the members of the proton pump, which sequesters the sodium ion into the vacuole at the cost of the proton hydrogen ion, right? And this ultimately also energizes, it creates actually a potential gradient across the plasma membrane or the vacuolar membrane. So it also energizes the other, the antiporter genes like sodium hydrogen exchanger, sodium hydrogen antiporter, or the sodium potassium exchanger. So this is one of the key genes, just like the plasma membrane ATPase. We actually saw very clear-cut difference of the plants that overexpressed this specular ATPase from Spartina alternifera, be it in tobacco or in Arabidopsis, or in rice. So we can see here a clear-cut distinction between the wild types that are actually wilting and drying in 3 days to 1 week after salt stress, and then the overexpressor lines are still maintaining good bit of relative water content to maintain to have the good tissue tolerance. And also at this reproductive stage, and these plants, when they were actually moved to the greenhouse, at the reproductive stage, you can see a clear-cut difference between the wild types that do not have that gene and the overexpressing plants over here. Okay, so we have actually a lot of genes we have overexpressed and then seen some very good response. And this is another gene, ADP ribosylation factor. It's actually a small GTPase family protein, and it's actually more involved in the trafficking. So we saw also that very clear-cut distinction with Arabidopsis and rice. So here you can see also it's not only at the above ground, but also at the root level. We see very clear-cut distinction between this the wild type and then the transgenic overexpressor lines. And then not only just in salt stress, but also in drought stress. So when we looked at the drought response of these plants, we were amazed to see. And then, well, we knew that the extremophile genes will be also contributing towards drought tolerance, but we did not see that one with the vacuolar ATPase. Maybe it is more specific to just salt stress. But with this ADP ribosylation factor, we found actually very good response both at the above ground and then the below ground root tissues. Another gene, as I told you, that one more ability of the plant has to accumulate a lot of osmolytes, compatible osmolytes. So here is actually one gene myoinositol phosphate synthase. I know a group of Arun Lahiri Majumdar in Bose Institute. They have done a tremendous amount of work with these inositol pathway genes, and this is one of the examples of that. I mean, one of the genes in that pathway. So which leads to the myoinositol biosynthesis. And there also we saw a very good response of the salt stress in Arabidopsis and in vitro and also in the pot level studies. Okay, I would be spending a little bit of time on this particular gene because this is very close to my heart, because the response that we saw with this gene in Arabidopsis as well as in rice was overwhelming. So we actually went ahead and then patented this gene. We were lucky to have a patent on this gene because until 14 days, even the plants were not showing much symptom. Of course, the growth was definitely a little bit slow, but the plants were not showing much symptom of drought, whereas the wild type was struggling. And then when we put them back into water, the wild type completely died, and this survived as well as it went up to producing the seeds. And this is a wild type which we actually took out from 7 days of stress. The one that we had at 7 days, we took out one of the plants of wild type because we know that at 14 days it was dying. So we have 7 days that recovered, but then you can see the difference between these. And these are all actually even greenhouse and pot scale studies. So we have not done any field studies with any of these transgenics. If need be, or we definitely are looking towards doing that as well. So this is also again a very high root biomass. So we know that this root biomass is actually contributing towards the drought tolerance of these overexpressing plants, because we know there are actually genes for deep rooting that are contributing towards drought tolerance. So we are thinking that it may not be none of these varieties have DR01. I mean, Nippon berry doesn't have the DR01, but the one allele that is actually contributing to drought tolerance. But then you can see here that this gene is definitely contributing something towards the root biomass. And here the float leaf assay with the salt, under salt, clear-cut distinction between the wild type, under salt, and then the transgenic overexpressors. Here also in the hydroponics, the same thing. So we wanted to look a little bit more towards the mechanism of this gene. Okay, so we looked at actually under mannitol in mimicking the drought stress or osmotic stress by providing mannitol, and looked at the actin structure, because this particular gene actually is an actin depolymerizing factor. So what it does, we all know actin is actually one of the major components of the cytoskeleton. So when under stress, the actin is a very dynamic filament, right? So it goes from filamentous stage to the ground stage, the F to G, and that F to G interconversion is actually a dynamic process, and that process is somehow compromised under the stress condition. So because you need to have a constant supply of the polymerization of the G actin to the filamentous actin, so that there will be depolymerization to have the G actin accumulation. And we looked at the actin filament structure of these overexpressed plants. So the thing that I did not tell, one thing is we actually overexpressed the homologue of this particular gene from rice. So we overexpressed the rice gene in rice, and then the spartina gene in rice, right? So we are looking here, this is OSIDF2. That means this is the homologue of the spartina gene in rice. And as you can see here under control, these are actually the cortical cells. It's not like typical protoplast, but we try to extract the protoplast. It's very hard when you actually fix the tissues and then try to extract the protoplast, you would be expecting a lot of tissue breakage because they have been fixed. So we were not able to get the real, spherical protoplast that you have seen. That is good from the live cells or live tissues without fixing. When you fix it with some chemicals, then it actually loses its structure. So we looked at the ones that we got from the cortical cells, and then you can see here under control condition, then the actin mesh is kind of very intact. But when you put it under stress, the sputina is still kind of, the actin mesh is kind of still better than what you see here in rice, like it's much more chopped type, like it's fragmented. It's not really like depolymerized, but it's like fragmented. I'll show it in a couple of minutes what I mean by that. So you can have the actin depolymerizing factor would actually depolymerize, but if you see actually a lot of fragmentation, that means it is actually the impact of the stress, that it is not actually the G actin, but it's actually the fragmentation of the filaments. So we looked at that. So this is not actually a gene, that is not a transcription factor, or it is not, so it's not a kinase. So it is definitely not something that would be responsible, because when we did actually an RNA-seq of these transgenics, we saw a lot of kinases are actually being regulated by this gene in the transgenics. So that actually led us to the hypothesis that maybe this gene is not itself a transcription factor, but it's probably working actually as a transactivator. So we actually tried to prove that one by seeing the protein-protein interaction of this gene with few genes which are known in the interaction network. For example, the calcium-dependent protein kinase and the adenine cyclase, or the WD40 protein, or even the calmodulin receptor-like kinase. So you can see here, this is with sputina. In sputina, wherever you see here for calcium-dependent protein kinase, the signal is very weak. But in rice the overexpressing plants of OSIDF2, the rice gene overexpressing plant, is actually having a strong interaction with the calcium dependent protein kinase. So I would tell you why this difference is. When we went to look at the structure of this gene, we looked at the structure of this gene from different plants, rice and sputina particularly in here, and also we have done with some other like potassium and others, but here it's just rice and sputina, for example. We can see there actually in the 6th position, there is actually a change from serine to threonine in sputina, but in rice it is serine. Okay, so we know that the kinases, they will phosphorylate serine much more efficiently than threonine and tyrosine in that order. So we thought like probably because of this phosphorylation, so the rice gene is being phosphorylated higher by the calcium dependent protein kinase, whereas the sputina gene is not phosphorylated to that extent, and that is the reason we are seeing that the interaction of the sputina gene with calcium dependent protein kinase is actually weak. Okay, and this is actually also another 3D structure where you can see here that the rice gene has an F loop. This is the loop which is involved in the actin binding. And then in sputina, that F loop is much more protruded and much longer, this one. So we also thought that this is possible because this has a much more higher actin binding capability. So the sputina gene has very high actin binding capability, whereas the rice does not have that, because you can see here very minimal protein input, that the binding of the sputina gene, sputina is much higher than in rice. You cannot see here in the very minimal input of the actin, but in sputina you can see very minimal input. Also you can see the actin co-sedimenting. So we know there is a difference between the actin binding. So that means sputina gene is actually binding more, or at least facilitating more actin binding under stress. And because it is binding, so obviously it is depolymerizing. And we know that the activity is probably more because it is less phosphorylated because of that serine 309 substitution. And we did have in vitro studies for that to prove that yes, phosphorylation that the spartina is phosphorylated less compared to rice. And when we actually mutated in vitro that serine to 309 in rice, we can see, yes, in the rice gene, when we actually mutated the sixth amino acid from serine to 309, you can see here actually again the phosphorylation reduced actually, So, and there is another one at the 132 position, but that actually did not. So we actually we knew that this particular sixth position serine 309 change is actually contributing to the more efficiency of spartina gene. Okay, and these are actually the real-time actin filaments. You can see here this is the total reflectance microscopy. So here you can see the actin filaments, and then we took a time scale measurement of the actin filaments. In the slide you put the actin depolymerizing factor protein, and then with purified actin, and you can see actually this actin depolymerization real time. So I have probably a video for that just to show very quickly a time scale actin depolymerization. Okay, so here it is. This is actually the, how do I do that? I cannot seem to have the video running for this one. Is it not possible in the sharing the screen or?
No, sir, it's work, but I don't know. Is it the video file or it's
No, it's a video file. What do you call, like MPG or?
Okay, animation. It's on maybe.
No, it's not animation. It is a real time.
Okay.
It's a picture from the microscopy. Let's see the pointer. I'm trying to see the pointer of some.
You may choose
No.
the laser pointer.
No, that's not, doesn't seem to be working. Okay, but anyway, what I wanted to show here is if you see it real time, I mean, you can see it within 20 seconds, the rice actin filament gets, it's chopped. It is not really like depolymerized. And we actually are targeting by using, this is the actin has been pyrin leveled, and then you can see but we are also, this is only specific to the F actin. So we are also now labeling the G actin. So we can see real time both F and G actin as the depolymerization continues, and then there is accumulation of more G actin. So unfortunately this video is not working. So, but anyway, let's just move on. So I would like to conclude my talk over here. The bottom line is, these two grasses are not really like crop models, but they are actually wild relatives of crops like rice, for example. So an integrated approach of studying these monocot halophytes such as Patria. My focus is now, my laboratory more focus is on Patria. Because Patria is a wild rice and also genetically much more simpler than the Spartina alterniflora. Although we have done a lot of work, but then we can translate that information in Patria. And now that we have a very comprehensive transcriptome atlas, and then the genes that we have identified from the functional screen. So we hope to make more and better progress with that. So studying those, for example, Patria at a systems level would not only just increase our basic understanding that we know the stress tolerance is like a puzzle, right? So everybody has their pet gene, everybody promotes one of their genes, and that's nothing wrong. That's good, because it's just like a crossword puzzle. So we are just like adding one puzzle to that one piece to that puzzle. So we all are contributing little bit to that puzzle. So to have a better and clearer idea of this stress response, and then once we understand that, then it will be easier for us to improve the stress tolerance. And I did not show again because it's not published. So we do have some lines that we have developed by gene editing, because we know some of the key regions that are actually, for example, like that serine to threonine substitution, right? So we do have some base edited plants that are, showing some very expected response to drought stress and also salt stress. So that's having said that, I would stop here, and then I hope that I did not go a lot late. So and then with that, I would like to acknowledge these wonderful people that I have or had. I know bunch of the work was done by Venkata Mangu. He is my postdoc, long-term postdoc. He was 6 years with me. He's now actually working for a company in Missouri. So it's a company that works with chloroplast genetic engineering. And Sonali, who was postdoc with me for 4 years, more for basically all of the protein works that I showed is her contribution mostly. Mangu was involved in pretty much all of them. He was like I call him like my right hand. So he's like pretty much like a plug point. You can just put anything like an adapter. So, and Rohit was a postdoc with me for a kind of short time, 8 months, but he was very productive. I'm glad that he is now a senior scientist at Himachal. Himachal Pradesh, right? And then I had a visiting scientist, Nezla, who actually screened those overexpressing lines for drought stress, particularly the functional screen from Patria. Jonalyn is right now, she's more like a right hand to me now. So she is a research associate with me, dealing with rice work, and then also helping me, all these lines that are developed. And Mohit is my current informatics guy, bioinformatics guy, who helps in analyzing all the next gen sequencing data or anything that has to do with networking and whatnot. And Nia was my research associate, but she had to leave, unfortunately, to Philippines for some family issues. But she's the one who was involved, who actually developed the gene edited plants. So, and then we are continuing with that. And Eduardo was my former student. He initially started working. He's the one who was initially with Mangu Ramana. I call him Venkata Ramana Rao Mangu. So I call him Ramana. So Eduardo and Ramana were heavily involved in the salt and drought stress tolerance of the Spartina genes. And Renes was my former, and Eduardo is actually a professor now in Ecuador. Nezla is a scientist in Turkey. Renes Bedre did a bunch of those the bioinformatics work that I presented. He is actually a research scientist in Texas A&M and West Laco. And Dishna Ratnasekara, I must mention that because she was only 3 months in my lab. And it was a very good coincidence because we were actually screening those rice plants for salt tolerance, and then she could fit into that. And then she did a very very good job in that 3 months to warrant a publication. So and these are the people that have contributed to most of the work that almost all the work that I presented today. And there are many others who have worked just very short term, maybe helped in this as Borlaug fellows or just short term trainees. And I have a very big group of collaborators, Andy Pereira from University of Arkansas with my rice work, and Rodwing with the Patria work, and then Dr. Rajasekaran from USDA. He's just opened his lab for me, like anytime our microscopy studies in addition to our microscopy facilities, and also the tobacco work is contributed from his lab. And these are the people with the rice screening work, mostly for the aerobic or drought screening. And I did MicroRNA in Dr. Galbraith's lab in Arizona back in 2008-9, so that deserves to be acknowledged. And also the scientist of the coastal plant group in LSU, and because that is where actually I started here as a postdoc in the coastal plants program. So there are some scientists like Dr. Harrison, Dr. Subudhi, among several others that also you know, need to be, or I do acknowledge and then appreciate their help and then support. And of course, none of this work will be possible without money, funding support. So, the funding support from USDA, National Institute of Food and Agriculture, USDA, and then the Louisiana Rice Research Board for some time, and then of course my institute, LSU Ag Center. So with that, I would like to what is this? Want to keep your link and not as
yes, sir. You can. You
stop
yes,
share.
yes, yes, sir. You can stop your screen. Sir, there are some questions. We gather so many others too, but we try to put few of them. There is a option in the chat box. You may find.
I am looking at, so from Ratna, are these selected genes expressed in all stages of plant against drought? No. You do not want the genes to be expressed at all time points and in all tissues in the plant, because that would have some phenotypic cost, right? So you do not want to express the genes. Yes, the transgenics we have actually expressed. The overexpressing transgenics we have used either ubiquitin or 35S because we wanted to express in all the plants and in all the tissues, and then so we can actually know which stage, because we are screening the plants at different stages also, right? So we are screening for salt tolerance at the germination level, at the seedling level. So if we actually use a tissue specific promoter, then we probably would not be looking at these plants showing stress response at the seedling stage at the same time at the germination stage, So in the overexpressing plants, yes, they were actually expressing at all stages of growth. And that is the reason probably we have seen some growth retardation of some of the transgenics, which is expected. And once we identify the genes, like through this gene hunting that we call, and then we can actually have the tissue specific promoter or some promoters specific for a time point to overexpress those genes. So it's a good question. Yes. Can any plant growth promoting rhizobacteria have any effect on salt tolerance? It's a good question. It is more and more evidences are accumulating that yes, it's not only just bacteria, even the mycorrhizal like arbuscular mycorrhizal fungi are arbuscular mycorrhizal fungus, right? So they are also contributing towards stress tolerance. My group, we have not done anything on that front. I would just cite or quote the published literature, and then that actually occurred that yes, it is true that some of the beneficial bacteria, so probably rhizobacteria is one of them. So can we introduce a transgenic gene in rice against salt stress for the future yield productivity? Okay, can we introduce a transgenic gene? Okay, yes and no. So the answer yes is because can we? Yes, we can. But is it going to be marketed? We don't know. We still do not have, we at the United States have, I think, probably maximum number of GM crops are being commercialized here, right? So we have GM cotton, we have GM soybean, we have GM canola. So, but when it comes to rice, there are different things with GM rice, because rice is sacred to many, and rice is also one of the export commodities. So you don't want to mess up. For example, US rice export markets are in Europe and in Japan and some other countries, and they would not really entertain GM rice, So we can, but then it is not. So that's the reason now we have the ability to edit, those genes. We look at the gene structure and then find out the targets in that gene, and then try to edit, because at least in US, I know in some other countries they are also working on it, but in US at least the gene edited crops are deregulated, so, and we hope that that's the same case in European Union. I know they have not really deregulated, they are still regulating just like the but then we really need to know whether it is actually CRISPR-Cas9 or base editing, or so how the gene edited plants will be developed, and then how they are going to go about it, But yes, it can be, but you don't. Now in the era of genetic editing or gene editing, we are not really talking about transgenics directly going to the field, unless and otherwise there is something like golden rice, which is probably you don't see a lot, although there was one report from the Pam Reynolds group that they have edited some genes for producing golden rice, or the beta carotene rich rice, by gene editing. But until that comes in, so the golden rice, the original golden rice, still is moving forward towards commercialization. Yeah, if I'm not mistaken, I think it's in Bangladesh. Is that right? Bangladesh and Philippines. So they have gone one step closer to commercialization. So I think it's moving. That was a very special case, and then it should have been long time, but then finally we are happy that it's moving. But when there is an opportunity for going for gene editing, then we must and we should, because that's free, and then that's safe in terms of regulatory restrictions, So but you do not want to use the transgenics just for producing developing a product, because gene editing is also a process which involves transgenic technology, right? So the technology per se is transgenic, right? You still transform the plant and you still produce, but then the product is just not transgenic. So you would use the transgenic technology to validate the function of a gene, and then you can actually know that yes, this gene is actually doing something better, and then you look the structure of that gene in versus susceptible cultivars, and then see what are the target areas of that gene that you would like to edit, and then what strategy you would be using for that editing. In the transcriptome analysis of quartet, which genes are showing differential expression? Okay, yeah, I did not present that one, definitely. So, but we looked more of the functional screen part, I can tell. I have not really talked about the transcriptome atlas since it's not been published, but we have seen similar things like more on transport, and also on the metabolic pathway that is leading to osmolyte biosynthesis, and also the genes. There are a lot of genes in the kinase, signaling pathway, that we have identified. So again, once we make a concise list of those genes, I would not be able to direct my answers to that particular question. But what we have seen in the functional screen, we have seen genes in kinase, signaling pathway. We have seen the genes also in photosynthesis, biosynthetic pathway, especially in the PS2. So yes, the genes are all over. So it's not just only a specific pathway, but again, it is looking, when you are looking at actually the tissue tolerance, we are actually looking more to the antioxidant pathway and then the osmolyte pathway. But when we are looking at something different, the root growth, then the pathways are different. So that is the reason I told that it's not just only one or two genes involved in just making a plant tolerant. So even if I am overexpressing one gene, it's still interacting with the genes that is already there in the rice to have that effect. It's not just that I can say this particular gene is actually just a master regulator. What could be the better methods to analyze the salt stress of rice?
It's very difficult to answer.
So I know there are different ways. Let me tell you that. So you can do it at the germination stage. Like, you can put salt water for germination. That is first step of screening. But just so you know that again, like, the salt tolerance is, it will vary at different growth stages. So the plant that would germinate well under salt may not really grow well under salt. Because at the germination level, there are some enzymes like peroxidase, dehydrogenase, those are actually more active. So if those are overexpressed because of your gene, then they might actually germinate. But then they may not actually hold good for your seedling stage, and then so to your reproductive stage. So if you want to really develop a salt tolerant plant that would yield well, you have to screen at all stages to make sure that it can germinate, because in the field, when you actually put the seed, whether it is direct seeded, then you have to have the seeds germinating and then surviving and then producing yield. So that is, but for the laboratory scale, we find most easy or more convenient is the hydroponics, because you can screen a large number of plants, and then the germination level also. So, but I know IRRI has, or maybe some other countries, wherever, I'm pretty sure that other countries, they have some plots like micro plots they call, and then they actually kind of irrigate with salt water, and then they are actually constantly being saline. So maybe that's actually a good way of screening your plants or your varieties. In rice seedling and reproductive salt tolerant genes are different. How come these genes give tolerance to both stages? That's good. So I'm talking about one gene that is giving at reproductive stage. We didn't test for reproductive stage stress in all of that. There were vacuolar ATPase, because that is actually a mechanism which is operational at any stage of the plant. Okay, so and then there are some of the genes, as I said, if I could go back to my slides, or when you go back to my slides, you will see that at the vegetative stage, I had a lot of Arabidopsis plant and rice plants, not rice plants, Arabidopsis plants. They were actually tolerating. And when we went to the reproductive stage, there were only 5. So the ones that were giving tolerance at the vegetative stage did not make the cut at the reproductive stage salt stress. So you are right, they are different, the genes are different, and that's the reason those plants which survived during the seedling stage, not all of them made it to the reproductive stage stress. Okay, I do not see any other questions here.
No, sir, it's enough. It's enough.
Okay.
And we have collected others' questions, but we think that you have all covered in your talk. Maybe they come at later stage or in different time and missed it. So
Yeah.
I'm request Shoma to do a interview to our speaker. Shoma, please.
Hello, everyone. Okay. We are now moving ahead to the interview section of today's webinar. Today is our 19th webinar of the series, and we are overjoyed to have with us Dr. Niranjan Baisakh join us from Louisiana State University, USA. Sir, it's wonderful to have you here. And it was a wonderful presentation, and your talk was appreciated by so many people. We were watching the chat in YouTube. Sir, we would love to know more about you. And this section is all about that, to know more about the speaker. And we are very interested to know how you build your career. Could you please tell us about your journey so far as a plant scientist?
Okay. I as you as um, Liberty said that I did my undergrad in Orissa University of Agriculture Technology. It's in the eastern part of India. Let me tell you that if you really, some of you may not be aware, Orissa, the name actually comes from Orissa. Okay, and that's the etiology of the name Orissa. So because there are of course difference in the opinion that rice, the primary center of origin is actually Jaipur. That's actually a small city in Orissa. And until so then of course there is a conflicting report from the Chinese counterpart. So that's the reason they say like Asia and Asia Minor as the center of origin. They don't say the center of origin, the center of maximum diversity of rice. Okay, so I grew up actually in a rice belt. In Orissa, of course, we eat almost, I mean, every day 2 times rice. I would not go to the bed without rice in the night for sure. Uh, so that's how important rice to us, right? And I grew up. My father was a small scale farmer, so we go help in the field whenever it is needed. And during those days, we used to compete with our siblings or our neighbors, because the rice were not very developed varieties like the improved varieties. So most of the panicles will break also. So what happens, because and this is not like mechanical harvesting. So I'm talking about maybe in 1970s, so they were still using a lot of those land races or those that time so-called improved varieties. And then we will go after it is harvested, the field will have a lot of broken panicles, so we'll go with a basket, and then whoever can fill that basket and comes home gets some money from the parents, so that's how that was actually our after school kind of running to after school and run to the field during the November December time. So it's not that I'm not going to tell like from the childhood I had the interest. No, I really wanted to be a doctor, a human doctor. Okay, so after my higher secondary, I wanted to be a human doctor, but I was in the waiting list. I'm still waiting. But since I could not make the cut while waiting, so what I did is I went to the agriculture streamline, and my father looked at me. He said, "Are you really going to put a plow in your shoulder and then plow the land because you are going to study agriculture?" I said, No. That's not really how this is. We'll be studying about a lot of things about agriculture, soil science. We'll be studying about the plant varieties, and then so I became very, very interested in genetics. And that credit actually should go to Dr. Chittaranjan Kolle. He is my master advisor, but he was my teacher of genetics and plant breeding in undergrad. So I just cannot imagine the way, or I would try to emulate him the way he teaches. He is probably the best teacher that I have actually come across in my life as far as genetics and breeding is concerned. So I became more and more interested after his classes and interacting with him. So I'm one of those few ones that time that got involved in doing research as an undergraduate student. I know this is a common trend here, but during that time, in 1980s or 86 or 87, so I was looking at working with him in the field. He was doing some and the citronella lemongrass thing, and then he would take me to the field and just select. So that is how I developed my interest in research. That's the first step, that I developed my interest in research. And moving on to my master's, I worked again with him for my master's thesis. So I started to work on tissue culture, because tissue culture was the biotechnology that time. you know.
Mm-hmm.
So if you know, and also it's more, you know, kind of fancy because you are staying in an air conditioned room, you know. So you consider yourself as an elite group of people, students, you know. You are in an air conditioned room because tissue culture has to be in the air conditioning, you know. So everything, so that's the reason. Anyhow, it's not, that was not the reason that I was interested, but I was interested in tissue culture because that was the biotechnology, emerging biotechnology, you know. So I actually stepped into the tissue culture process. I started working tissue culture of first thing was with the peppermint, you know. That was my first tissue culture adventure, because our idea was, that time we were still kind of brainstorming what to do, what is the tissue culture to do, because my university doesn't have a tissue culture facility that time. So Dr. Kole made a connection with a scientist at Regional Plant Resource Center in Bhubaneswar, India, and where I went, and then they had a tissue culture facility. So he just thought, like, you know, Dr. Kole has tremendous foresightedness. He said, maybe we should produce menthol in this. I mean, menthol is a secondary metabolite, right? So we'll produce menthol in tissue culture medium, you know, like in bioreactor, small scale bioreactor in class. So that's when I started to work with menthol, peppermint. But then after a couple of months, I thought it was a very difficult journey for me, because I'm studying in one university and then going to another place. And there was, you know, that time we did not have public transport that good, and I did not have, so I have to go by bike. So then one day I told that, you know, I don't think that it is really a good idea, and or I can do much over there. And then these resources were limited, so I didn't have money. I was just getting enough money scholarship to pay for myself. So he said, you know, I can support some of these supplies, so that's okay. I still remember now, I went to one of my university staff. This is a funny incident, but I went to get some sucrose. So the technique, well, you know, he had one bottle of sucrose probably, maybe, I don't know how old. So he just took out actually a paper, like a newspaper piece, and then gave me. I said, no, I'm not asking this sugar for tea, you know, I'm asking for preparing some media. So I need some 30 gram. He said, what? 30 gram? No, that's not possible. There are other students also, so it's not possible. So that's how we had to grow up. So so I told to Dr. Kole that this is kind of tough, and when I'm going to that institute, they are not very open, obviously, because everybody is limited with their resources. So he said, okay, let's just think, and then, you know, maybe, but I want you to do tissue culture though, because that's one area that he was deficient with. So he wanted to make the team complete by me making the tissue culture progress, you know, progress in tissue culture. So then we somehow talking, then we decided that, you know, this is a crop that I had interest, that Lathyrus, called Lathyrus. It's a Lathyrus sativus, you know, it's called chickling pea, veg pea, and it's called a poor man's legume. It has very high nutritional value, very high protein, and very rough crop. Like it can grow in very marginal soil with very limited or attention towards nutrition and all these things, so even water. So I thought, like, you know, this is the crop which is banned because of the neurotoxin that it produces. So even though you have to eat for 17 years to develop that paralysis symptom, but still it was banned by the government of India, that it is not for human consumption, because it does have that it's called like boa beta oxallyl beta n oxallyl amino acid, so alanine amino acid, so it's called boa or dimboa, and that particular neurotoxin was being produced. So we thought, like, we can produce some somaclonal variants that could have less of that neurotoxin, you know. So that was our idea. So I started doing tissue culture of Lathyrus. And believe me, the tissue culture of legumes are definitely difficult. And during that time, so and I know one scientist, Dr. Srinivasan at Indian Agriculture Research Institute, working with chickpea, and he was having terrible time too. So he said, you know, legume tissue culture is very, very tough. And we, obviously I had tough time. So I worked actually in one university, Utkal University. I used their facility, and then there was good communication of the city bus, town bus we call. So then I could go, and slowly Dr. Kole saw my progress and became happy. So he kind of lended his scooter to go there, so that I can save more time by going there. So that's how actually I started into my journey into tissue culture. Okay, and then after I graduated with my master's, I really wanted to continue with Dr. Kole for my PhD, but he went on to Wisconsin, University of Wisconsin, and then he stayed there forever, almost like 4 or 5 years, So I was waiting for him, and then I decided that I need to move on. So I actually, rice is not new to me, and there was actually a research scholar position open at Central Rice Research Institute in Cuttack. That's actually our Indian Rice Premier Institute, or now it's called National Rice Research Institute. So my supervisor then, Dr. Jijan Rao, who actually returned from Texas A&M and was establishing an anthroculture program. So, and then that position was there, and every time I pass by that institute, I always think that I should work here. So, and there was this advertisement, and then I met with Dr. Jijan Rao. Even that advertisement was not there, out yet. So I had a couple of friends working there in breeding, and then I visited them, and then they took me, and then they said, "Oh, sir, he has some experience in tissue culture." He said, "Okay, we have some position coming in maybe." So, then I applied and I got it. And that's how actually changed my whole perspective of, the crop. So I decided that, no, I will be sticking to rice, So, and then I did a lot of anthroculture work with him. We did very, very significant progress with anthroculture, double haploid, like we produced like hundreds of double haploid, and Dr. Jijan Rao, and that was actually carried by my successors in his lab. And then he went on to even develop some varieties from those, those double haploids. But I got tired because I am a person who always wants to learn, and ever learning person I am now. I cannot just imagine that I can be stuck in some time and then I don't read or I don't do something. Even now also I'm a hands-on person. I still go to the lab and then do whenever it is needed or whenever I would like to. So I'm more a hands-on. And I said 2 years later, I told to Dr. Rao, sir, this is too much. This is like becoming more like monotonous, mundane. So I really want to do something, because the idea was to develop some protoplast derived transgenics, and that was the project. Where is that? He said, "Oh, well, we don't have the facilities here to have the protoplasts, and then we don't have an inverted microscope and all these things." But there was a Rockefeller Foundation grant actually during that time. Rockefeller was pouring a lot of money for prioritizing rice biotechnology in Asia. So that was actually, so a lot of money were there, but we don't know, at least in my lab there was not a lot of money, that we didn't have any microscope. So I owe to Dr. Rao this, that because of him I got the opportunity to see outside of India. So it was one conference in Delhi. It is called National Rice Biotechnology Network. That was a meeting. It was sponsored by the Rockefeller Foundation. So basically, in that place, all the PIs, the principal investigators that have been funded by the Rockefeller Foundation, they will be presenting their results. So that is where Dr. Rao allowed me to present my work. And then and Dr. Rao actually, because he was a Rockefeller Foundation postdoc at Texas A&M, so he connected me to Dr. John O'Toole. He was actually the coordinator of Rockefeller Foundation for the South and Southeast Asia. So Dr. Rao told to John that, John, this guy has done this work, and John O'Toole has already heard me. And then during the lunchtime, he kind of tapped on my shoulder, and then I said like, okay, who is that? Because I was afraid that last night maybe I drank a little bit more, so maybe he's telling me something. So this is the truth, because we were in Orissa, we don't have access to a lot of good things, and then when in Delhi we saw like a bunch of those things, and we are very young. So then I said, somebody is trying to tell me something, So, and then I looked, and then tall man like John O'Toole he was. So he said, young man, what is your next plan? Then I said, no, I'm going to attend to some talks. And then he said, no, I'm talking about what is your next plan in your career, what you want to do. And I said, oh, then I didn't know he is John O'Toole. So I looked at his base. I said, John O'Toole Rockefeller Foundation. So I said, okay. And then Dr. Rao had given me an hint that I would be talking to John. So he may talk to you, So, but I don't know when. And that's when actually my life changed, because he said, so what's next? I said, well, I'm kind of tired with this anthroculture. I'm thinking to do some more transgenic work. And then he said, so do you know where? I said, we don't have that facility, so I don't know where. He said, do you know there is a fellowship given by the Rockefeller Foundation? I said, no. He said, okay, go back and then see in the backside of, they used to have a Rice Biotechnology quarterly magazine by the Rockefeller Foundation. So go back, and then the last page of that will be a form. Fill that one out, and then apply. So that was it. So I did go, and I applied with my this thing, because there was no internet or nothing for that matter. I think the whole CRI had only one computer that some people used to use for something, maybe. And then so when I sent it, and then I sent my CV, so-called CV, in my hand, because
handwritten.
in my handwritten. No, it's not CV, my handwritten. My CV was typed by Dr. Rao, but I had my research
Application.
accomplishment.
Okay.
Research accomplishment, what I have done so far. So I wrote it in my hand.
Okay.
And John O'Toole visited, so this was in November 1996. John
Okay.
O'Toole visited CRRI in April of 1997.
Okay.
So he said, young man, I'm interviewing now the candidates of in Asia. So you are one of those 60 or 70. So I would like to talk to you sometime after lunch or during the lunch time. So we learned that he's a vegetarian. So I'm a vegetarian. So we were in the same table, because in the lunch time. So then he said, and he opened his laptop. He said, have you seen this form online? I said, online? No, no. He said, that's very strange. And he showed to the people my handwritten that paper. This guy has applied to me in a handwritten thing. So anyway, he took my interview. He asked me a bunch of questions about what I plan to do in life or whatever. And then he kind of said, like, okay, that was in the lunch time. In the evening time, before the social, he said me, okay, I have forwarded because while he was talking to me, he was doing something on his laptop. So he said, like, he's filling out some forms. So, and he said, I have forwarded your case to the main office in New York, Rockefeller. It's in New York. So, and that time Gary Tonison was there. So he said, Gary Tonison is here. So I will talk to him. And then, you tell me where you want to go. So I said, immediately I said IRRI, because I'm working with rice, and no better place than IRRI for working in rice. And I had very little outlook. I really didn't know that rice is also being grown in US, not much. So didn't do those kind of searches much or something. I know research is going on, but I don't know that rice is really grown in this scale or, So then he said, why IRRI? And I said, well, IRRI is Premier Rice Institute, and then it is in Asia. It's close by to India. And then it's an introvert person like me, so I don't want to go too far. And Dr. Datta is a pioneer during that time, So having come with this fast protoplast transgenic fertile rice, and then a lot of accolades for that one. So I said that would be an opportunity for me to work with him, So then he said, okay. And Dr. Datta was there. There was another meeting immediately following the NRBN called the Crop Science Congress. So the Crop Science Congress was in actually science in the Bigyan Bhavan. So that was just immediately after that NRBN meeting. So that is when I met with Dr. Datta. Then and then John O'Toole must have talked to him, and then Dr. Datta said, okay, so you want to do PhD with protoplast transgenics? I said yes. And then I said, okay, just write down a small kind of a proposal, 2 page or something, and then send it to me. And then, so that was like the life changer for me. So, and then the rest, I got the fellowship. I went in November 1997 to IRRI. And then after that, I have not looked back. And then I stayed there quite a bit of time, close to, I think 6 years, 8, 9 months I stayed with him, and different projects. First I did my PhD in rice, in agriculture, double haploid transgenics, and then both bialistic and agrobacterium was just starting, in rice. But if you look at actually the agrobacterium publications, I think the one in theoretical applied genetics with Karabimem as the first author, that was actually the first rice agrobacterium transformation paper.
Right.
So agrobacterium has been in several other crops, but not in rice per se. So, and then Dr. Datta was not actually a firm believer of agrobacterium. So I remember him vividly saying, when I showed my results to him, he said, it's a contamination, something like that. So, and then, so then I told madam how to convince him. Madam said, no, we have this before. So madam had a transgenic, Karabi madam had a transgenic with a maintenance line with agrobacterium. And when I had him little bit larger scale, so Dr. Datta said, no, this is not possible, because he was thinking PCR is contamination, because it is like that. He's from that old school thought that time, So, and it's nice because something to accept first, you need to really have a good bit of scrutiny, which he was doing. So there's nothing wrong in that. So he was trying to be more scrutinizing the results to convince him that yes, it is working. But once he was convinced, then there is nothing looking back. He stopped protoplast, and mostly bialistic still continued because there are still some recalcitrant varieties, and agrobacterium was just starting. So basically Karabimem started with agrobacterium with a person from Switzerland, I think Stenaka Nikolova. I cannot pronounce her name. I should not be doing wrong to her name, but justice to her name. So after my results, then she also got more and more convinced. Then we put together the paper, and then that came out. And that's actually one of the first papers of successful agrobacterium transformation with rice. So yes, after that I was very lucky to work in the Golden Rice project, and I'm also one of those older persons to be involved with Dr. Datta and that group to work with the Golden Rice. And after that, a lot of students joined, and then went bigger scale. I still cannot imagine that I would learn HPLC. I had to go to India. He sent me to India for a week to learn HPLC, to analyze the Golden Rice, and then I went to National Institute of Nutrition. Dr. Shivakumar allowed me to stay 1 week there. And then I came back, and then we had a waters machine, and then Dr. Datta had a full-time person just to do, screening of the transgenics or the varieties for not only beta carotene but also for some other nutritional traits. So that is where my IRRI, and then when I just kind of thought like, it is time for me to move on, to look a little bit different, because transgenic as a technology I was enjoying with the different traits. But me as a person, I'm always looking forward to more independent thinking also, and also doing more of molecular biology stuff. I did a lot of construct making in Dr. Datta's lab, a lot of southerns, westerns, northern, but something to think more into the discovery platform. That was my idea too. So we did some work with abiotic stress drive genes in his lab, and then and we moved on. But then here, when I came here, I joined actually the coastal plants program, and the molecular program of the coastal plants group was being led by Dr. Subhadi. So I joined in there, but the objective was actually to look at the genetic diversity of this, And I said, hold on, I will do that, but that's not something a postdoc job, So I would be rather more looking into why this halophyte, or maybe cloning some genes from this halophyte, and then doing for some stress resistance, abiotic stress resistance, particularly because, where we come from, or I come from, like in Eastern India, most of our land is actually rain-fed because our farmers are very small or marginal scale farmers. They do not have the facility to actually have mechanized farming and then also irrigating as and when it is needed.
Irrigation, right, right.
So most of our land is just rain-fed. You just leave it to the rain god. So if it is too much rain, there is flooding in the rain-fed lowland. If it is no rain, then in the upland it is drought. Right. So that's how we have grown up with East India, and that's the same thing with Bangladesh and then other Asian countries,
right,
and some other countries. So, and then now more and more so I was always thinking of abiotic stress in my mind to do. I was lucky. I was in just contributing a very minor or meager to the Golden Rice project, but that was in the very primitive stage of where we are talking about 1.6 microgram, and we are very happy because the original was 0.6, but
right.
now we are talking about 37 microgram, looking like carrot. So that's an evolution. That's an ever evolving process. That's okay. So I just tried to focus more on abiotic stress when I came here in Louisiana State University. And now that Louisiana is one of the major rice producing states in the United States, so I'm very lucky to be here at LSU. And then I started working as a postdoc for a couple of years, then moved on as a non-tenure faculty to have my independent program. So, and that's when actually I was lucky to collaborate with people like Andy Pereira and then some others to have some funding to do full scale. And then I recruited my first postdoc, Ramana, who stayed with me for some time. In between, he went for a year to ICGB, and then he came back again. So so that is how actually I built up my lab over here. And then after that, there is nothing looking back, So, and then, obviously, when I was working with rice, then there was this opportunity for sugarcane. We had a sugarcane scientist who moved on as a full-time sugarcane breeder. So I actually tried to take that, or I kind of was given to take that challenge. Because of a position thing, because I was a non-tenure, so I was made a tenure track in 2013, and then my focus was shifted to some extent to sugarcane. So so my focus is more on sugarcane, some in rice, and then also helping a little bit the other breeding programs, mostly helping with their students, like wheat breeding program of Steve Harrison, or and collaborating with other scientists on maybe a little bit on cotton and corn, so and sweet potato, but most focus is on sugarcane and also on rice. So that's why sugarcane is a very challenging crop in terms of genetics. You all know probably it's very complex. It still do not have a genome sequence available, genome draft available for the cultivated varieties, although there has been one monoploid genome. So, but then, I enjoy working more in challenging areas. So, and then, I'm lucky that I'm here, and then I have the opportunity to work with these important crops that are very, very, very key to the state economy and to the people of Louisiana. And these are global crops, so yeah. I don't know if I really answered to your question. This is how
definitely. Oh, it was wonderful to hear. It's what a story, and it's so nice to hear that you are always thinking about the farmers at home. Whatever you're doing, you're thinking about the farmers, how to help them, how your research will help them, the farmers. So it's amazing to hear. Sir, I'm moving on to the next question. Along with food security, what are your thoughts on nutritional security?
I mean, there is no second thought on that. Nutritional security must be achieved, because we know very well a lot of people on earth, even though they have enough food, but they are malnourished by nutrition. So, take the example of vitamin A, for example. There are areas, so that is where I know I'm on air, but I must tell that if I have enough money to eat, I should not actually recommend what a person who does not have enough money to eat. I would let him choose whatever he wants to eat. Okay, so and this is actually I would follow that, because if I know something is scientifically right, I will eat no matter how it has been developed. Okay, so I know a lot of nutritional security. For example, vitamin A. There is no rice that has vitamin A, more vitamin A. So how do you integrate vitamin A traits or those three genes into rice? The genetic engineering is the only way, right? So that is why I'm saying, like, we should not be thinking about the methodology unless and otherwise there is some serious repercussion of that product. So you should select. But yes, coming back to your question, iron, beta carotene or vitamin A, so, and then zinc. You know now how important zinc is, right? We are talking a lot about zinc because of the coronavirus.
Yes.
So
Immunity. Yes.
right, so you supplement your food with zinc, or taking more zinc supplemented food, so, or zinc rich food. So there is no second thought about the nutritional security. It's just only food security is two part, one at the productivity side and the other one is the nutrition side. So they have to go hand in hand, you know. Yeah, research must continue on this, I mean, the nutritional security. And I like that idea of phytonutraceuticals, you know, like producing the nutrients. For example, there is a company, Patria Bioscience, produces human lactoferrin in rice seed, you know. And talk about the not only just nutraceutical, but also the vaccines or different medicinal plants, for example. Not the medicinal plants per se, but like I'm trying to quote the work of Henry Daniel from Pennsylvania, University of Pennsylvania. You know, he's producing insulin in lettuce plants. You know, so you just take the lettuce powders, the leaf powders. That is, he's encapsulating them, and then you take them, and then you have your insulin dose. So this is an area that it has to continue. I mean, it has to go, and then there has to be funding. Because it is very important. It's very, very important. Malnutrition, nutritional deficiency is important for the human health. And we all know human health is very important. Yeah.
Definitely. Okay, sir, according to you, which sectors of plant science research will prosper in the near future?
Undoubtedly, you all knew. We are all excited that the scientist in gene editing, that pioneered gene editing, got the Nobel Prize, deservedly so. So gene editing is definitely going to be very, very routine now. I mean, more than ever, it is going on. But then, it is actually the worst thing for sure. Wherever it is possible, that would be there. Coming back to, for example, rice, there are several different areas. I touched a little bit. For example, waterfront, right? So you definitely want to go for growing crops in minimal water. I mean, just like I'm mimicking the slogan that they put it in. I don't know if IRRI is the one who started this, like crop per drop, So you need to have crop growth per drop of water. So water is very precious now. As I said, some places we have more water, but the quality of the water is bad. So it's not just an abundance of water, it's just the quality of the water also. So we need to develop, and these are going to be worsening with the climate change, and climate change is something that just cannot be stopped outrightly. It is an ever going process.
Yes, we are going to have less water. Yes, in the future.
We will have less and less water, and also the heat tolerance. For example, night temperatures are increasing, and we all know the repercussion of high night temperature on the crop yield growth. So those are some of the areas that I know. For example, yes, gene editing is definitely going to be a very, very big area in the next decade or so. you would see more and more products becoming available, developed by a gene editing.
Gene editing.
Yeah.
All right. So the last question for today: what would your words of advice be to the new research fellows?
I'm also still new for many things. So there is no advice. I think these are all known. Everybody would tell you the same things, but in a different way. I have a 3D approach, both for research and also for the youngsters. For research, we have a 3D approach, right? We develop, we deploy, and we disseminate. We develop a technology. We deploy the technology to our product development, whatever product that could be, and then we disseminate our results to the public, be it a scientific community or the farmers or the growers, producers. For the researchers per se, this is for the research, but the researchers per se and for the youngsters are we alike. I mean, I'm considered, that means as old now.
No,
that
no,
I,
no, no,
no,
no,
I'm just
no.
I'm just telling that. So anyway, for all those who are in the research aspirations line, that again the 3D approach, a drive and a determination and a devotion. So you have to have a drive. I have to do that, and I want to do that. It's not like and then that doesn't just help, because I want to be a doctor, but I couldn't get into through that waiting list. But I went to the agriculture, but I didn't really persist on staying one more year and maybe preparing better and then go for that. So even though I had a drive to be a doctor, but I could not. I'm a plant doctor. I'm kind of happy
Yeah,
with
definitely
that.
plant doctor. Definitely
So
good one. Yes, a
that's
great
okay.
one.
So, but then you need to have that determination. You don't want to give up at any time, So that is very important. You first develop a drive, then you actually determine yourself that, no, I want to really do that. And then you have to devote time. You cannot expect actually a 8 to 4:30 job for research. Research is not a job. It's a passion. It's actually what you really wanted to do, right? So it is not a 8 to 4:30 paying job that you just do it and then go. You have to have that passion. So that is what I'm calling like devotion. You have to devote yourself to that work that you are doing, and then only you can do the justice. The success is not just what other people see it. It's good if it's something is other people perceive it, but success is what actually you introspect yourself at the end of the day. Did I actually do something to make myself happy? Is it really worth doing, or am I going in the right direction? So never give up. We all have the saying, we know that perseverance prevails. Persist on something, your persistence will prevail one day.
Right.
Maybe not today, but it will prevail someday. Okay, so I can tell you, coming from a place, I told this one to one of my administrative staff, and then she was laughing with something different. That I said, I came from a place where we didn't have electricity until I was 4 years old, and she could not just comprehend that how was your life then? So how did you go to restrooms or something like that, So how did you take bath or something like that? So these are some of the things, but I am so blessed. I'm so blessed and that way, and then privileged that, I am here where I am now. And but it is because probably all of the well-wishers like Dr. Koleb and my teachers and everybody who were with me that contributed to my success, and then Dr. Rao, for whom actually I came out of India, and then believe me, I never wanted to come to US in the beginning because I thought I had a very, very low outlook. I was very bad in geography. I did not know much about this. I thought like, I'm a vegetarian. I cannot just stand people, having or, eating some beef or pork or meat or something. So, but here I am, So, so this is how you evolve. So you have that drive and your determination, and then you devote yourself, and you will evolve the process, and then you will finally get where you are. And we all cannot just achieve everything that we want, but we achieve something, then we should be happy and be happy.
Right.
Be happy
Right.
with what you have, because just think about the people who don't have,
Right.
like, nowadays a lot of this WhatsApp messages, looking at glass half empty, look at the glass which is half
Perspective.
empty, right?
Yes.
Perspective. So that's yeah. So again, these are the 3D approach
Awesome.
I
Awesome.
myself,
Awesome.
and then I just tell that. So
The way you speak is amazing, sir. I'm just a big
it's
fan
just
of yours.
thank you. It's just from my experience. So it's just right from my heart. So
And so many things to learn from your talk, and in so many different levels. It was such
I
a
know I
insightful.
yeah, I realized that there are a lot of students, so I hope I did not miss or them in during this. That's probably typical for a laboratory researcher. I don't know.
No, I'm sure you have
Somebody who is exposed to it,
motivated
so
so many people.
yeah,
No, many would be really motivated. All our students and researchers, I'm sure. And we have so many positive feedbacks also. And let's end this interview session, and let's go back to Shubho. Let's see what he has to say. All right.
okay.
Thank you, Soma. What I can say, it's a wonderful talk, and we got 1,325 viewers for this session, and it's recorded and available in the YouTube channel. So we expect that more and more people will see the webinar and the interview section. And I'm very happy that I just wanted to say two things. One is the IRRI, which one is very important for your life. Like for the Bioingene team member, Liberty and Jacinta and me, we are actually going for the training at the IRRI in 2018.
Oh wow, wonderful.
Is RR2P, Rise to Research Production Training. So
Yeah, okay, yeah, I know that.
yes,
Yes.
so we are met there. We actually talked there, and we found that our mentality is a little bit similar, and we wanted to do that.
You
So
would
this is
enjoy that place. It's a wonderful place. A couple of my students also went from here, so it's very very wonderful. Philippines is close to my heart. So I spent 7 years of my adolescence there. So so and I have a couple of Filipinos always in my lab with me. So
yes. And lot
Sir,
of
Shubho is a fan of Filipino food after he went there.
I am not really sure about that. So
actually,
that's a story. I don't want to go there, but I'm not really sure about that. So if you go as a vegetarian to Philippines, I survived with rice and mango juice for some day. So that's because
I
for
just experienced
Filipinos.
sticky rice first time in Philippines.
Oh,
He's a
okay.
fan of pork adobo.
Okay, that's right. So
Wow.
for Filipinos, anything with vegetable is vegetarian during that time. So so,
So,
I,
and another
but I
thing
have a
is
very nice experience. Yes.
yes, sir. And another thing is the Sapan Kumar Datta and Karabi Datta, and they
Those two
both are
people played a lot of
yes,
positive role in my life, of course, in my career development.
yes,
So again, like here also, as I said, like once I moved here, then there are a lot of people like especially Dr. Harrison in the coastal plants group, and then Dr. Shubhadi was in the initial stages of my postdoc time, and also in sugarcane there are a lot of people in my group. I have an amazing group of collaborators and then mentors. So that's right.
and we got few personal congratulation and appreciation for your talk. Maybe they are your maybe their colleague, someone missed you in a sugarcane institute. Uh,
Who?
I missed. I copy the name. I missed.
No, I Sugarcane Institute. No, I don't know.
I don't know. And another is maybe you are the mentor for his or her career because he's mentioned you as a mentor for his
What's the
career.
name?
I
Oh, okay. Yeah,
so
okay.
there's a lot of messages.
Okay,
I
okay.
think we got about 1,000+ messages during the chat. So most of the appreciation and questions. So we filtered out. So we are very hurry to collect the data because it's went out when we stopped the session. It's a live chat actually. Sir, it's a honor for us. I also follow your, like you say, your tag paper, your work in different fields. Because when I look to cite anything, so it's come to your name. It's come to your paper many times for my PhD research and my current research. So your contribution is amazing, and it's I think our viewers are acknowledged. You covered the whole scenario from the beginning of the biotechnology, as you say, is the plant tissue culture, and now you are doing the best editing in CRISPR. So it's a long journey, and it's rapidly.
No, sir always did the trendy work.
Yes,
Whichever was in trend, sir was there.
yes, and the main thing is that
As a scientist, we always
yes,
evolve, right?
and
So we have
sir,
to
sir
keep up.
actually, yes, sir actually mentioned it that he loves to learn new things. So it's a driven thing when we try to that. It's also happened. I think it's also happened to others like me. I'm doing that another thing now. I'm doing another thing. But if, as you say that, am I doing the right things? Is a good question to ask ourselves to motivate in the field, because we know that in research there is a lot of failures, a lot of negative results and that thing. And so I add another thing is sir is still this life. He believe when he actual see the things. He not believe believe on the just data like you said that the PCR or anything. Sir also tell he also try to believe that whether it is okay or not, or what is the perspectives. So it's a wonderful session, sir. I like to say something with your colleague, Dr. Ed Mobara. Are you there, sir? If you there, then I request you to say something. If because you are new and you learn you see the team of Bioingene. We would like to know about you more. Maybe in future we can arrange a talk if you willing to do and give to the presentation in our humble platform. So, sir, with this note, we wanted to end this meeting. See you soon. When you got some new results, I think we expect you more and more time in this platform.
Sure. I would like to thank you all, even the viewers and also the team particularly, all of you, like it's now late 9 o'clock.
It's a 9 o'clock. Yes.
Yes. And then, keeping up with me for the last 2 years, 2 hours, 2.5 hours almost. So
it was a pleasure. It was such a delight.
And thank you for this platform. Actually, you guys are doing wonderful.
Thank you, sir.
That's very good.
Yes, we try to build this platform more efficient for researcher and students also, because sometimes our talk is so high that sometimes we want to do that. Okay, we are also try to build some environment like discussion sort of things. Or
yeah, maybe I should have done something little bit, but I just didn't have enough time. And then since my title was that way, so I thought like,
yes. So
okay.
Perfect
yes.
for
Okay.
it was great. It was for
Liberty. Is the time to buy?
Yeah, I have class soon.
Okay. Okay. Thank you.
All right.
Thank you, sir.
Thank you, everybody.
Okay.
Thank you, sir.
Bye.
Have a good day.
You too. Bye.
Bye,
You
sir.
too.